Okay, so today we are going to discuss about the
Hello,
second
sir.
component of your molecular motors.
Hello, sir.
Uh, yes,
Roll number 793 present. Sir, I'm trying to
I
book
have
you.
marked it this week. I have marked it.
Okay,
आपने
sir.
बीच में
Thank
भी
you.
बोला था ना? I have marked it.
Okay, sir. Thank you, sir.
Yeah. Okay, so if we're going to look at our cell does a like it performs a lot of functions on its own. Okay, there's a lot of automation you usually observe in a cell. So if you're going to look at a cell, it's kind of a miniature factory where you tend to have self-assembly units, where you tend to have janitors, where you tend to have transporters. Like there are different types of functions that are exhibited by our cell. Okay, so it's kind of a miniature manufacturing industry. So if you're going to look at the different types of events that happen, many of them do tend to take part in mechanical activity. Okay, like you take so-called spliceosome complex. Spliceosome complex का role क्या है? They do take part in detection. They do take part in cutting down. They do take part in joining up of so-called exons. Okay, same way you look at the DNA polymerase, completely new DNA molecule. Okay, and you take a look at the translation mechanism. So translation mechanism में हमने आपको एक वीडियो शेयर किया था जहाँ पे राइबोसोम्स के ऊपर बहुत सारे assemblies होते हैं and you see that the RNA chain moves ahead and that takes part in synthesis of your so-called proteins. Okay, so you have different types of mechanisms that usually takes place inside a cell, and depending upon what even we have different molecular motors available in nature. We have protein-based motors, we have DNA-based motors, we have RNA-based motors. Most of the motors that are available inside the cell are usually protein-based. Okay, so out of all of these protein-based motors, few protein motors do exhibit mechanical activity, and in that again we have two different categories. One are rotary motors, and the other one are so-called linear motors. So rotary motors are something that does the function by rotational activity. Linear motors are something that does not take part in any rotational activity. Rather, they will be acting as stepping motors or sliding motors. Okay. So how does this activity come into play? Let's look at it. First, we begin with the rotary motors. So in rotary motors, we have two motors to study. One is about the so-called ATP synthase, which is crucial for generating your energy molecule ATP inside a mitochondria. Mitochondria में ATP generate होता है. कैसे generate होता है? This is the motor that usually takes place. And not just even in the last we studied about photosynthesis. So in photosynthesis also you have the protons being utilized. As I said, protons are utilized to generate ATP. So this is the mechanism which is being followed. This is the motor available in the chloroplast that gives you the ATP. Okay, so that's one thing. And second thing, we have another largest of the motors available in this so-called bacteria. So these has different rotors available because of bacteria has different so-called layers in it. So at each and every layer you have this so-called arrangement of rotors, and these rotors they are one of the largest rotary elements available on the earth. Okay, so let's begin with the smallest of the rotary motors. That's ATP synthase. Okay, or ATPase is short, we call it as ATPase. So ATPase basically has two components, F0 gradient and F1 gradient. F0 gradient is a membrane gradient which has three different elements, A, B, C. Okay, so A is usually made up of 5 proteins of A. These 5 proteins of A will be having only two jobs. One is it provides the inlet for the entry of a hydrogen ion. At the same time, it provides the exit for what do you say, exit of the hydrogen ion. So you have two things. One is inlet and the other one is outlet. So both of them are provided by your A segments. Then you have a C segment. C segment usually will be varying from 10 to 14 number of proteins. Usually bacteria have 10, mitochondria have 12. Chloroplast might have more compared to the mitochondria. Okay, so here we are actually looking at a mitochondrial so-called F0 fragment where you tend to find C12 elements. See, this is a protein. So these proteins we have such kind of arrangements in like in 12s, numbers of 12s. Okay, so Now these 12 proteins will be doing a rotary activity. Okay, there is a sort of a mechanism that is being adapted by them where they exhibit rotations. Okay, and the rotation is dependent upon the concentration gradient of hydrogen ions. Okay, so where do we get the hydrogen ions from? We get the hydrogen ions from so-called electron transport chain, and from the electron transport chain, we have earlier discussed about metabolism, right? Metabolism में जब भी आपका sugar breakdown होता है, या fat का breakdown होता है, या protein molecules का breakdown होता है, we have certain mechanisms being coming up from mitochondrial events. We have a Krebs cycle followed by we have electron transport chain. So Krebs cycle में जितने भी factors आता है, all of those factors will be acting upon a group of complexes available on the membrane, and these complexes will be liberating hydrogen ions into the intermembrane space. Okay, and at the end of these complexes, we will be having this fifth complex called as ATP synthase. So this is where your energy generation basically happens. Okay, so we have already come across the parts of FO. Now let's look at the parts of F1. Okay, so this F1 is comprised of 1 unit of gamma, delta, and epsilon. Epsilon is a very short unit. Okay, and the B is interconnecting from periphery with both F0 and F1. Okay, so how does this rotation basically happens? Let me just share a small. Let me just share a small video with you all so that you can actually look at the animation. You can understand what working is happening. Yeah. So you can see from here, this is your mitochondria. So this is your outer membrane and this is your inner membrane. The inner membrane is folded in several places. Okay. So when you're actually looking at the inner membrane, kindly have a look. You have different types of complexes available on it, right? You can see you have a complex here, you have a complex here. So these are all the complexes that actually takes part in electron transport chain. So you have 1, 2, 3, 4, 5. You look at the fifth one. Fifth one is bigger. This is your rotary molecule that we're talking about now. Okay. So this is the rotary motor, and this is dependent upon these dots. What are these dots? These are nothing but the hydrogen ions that are pumped into the membrane space whenever we have a catalytic reaction happening. Okay, so catalytic of anything. It can be sugar, it can be protein, it can be lipid, or it can be any other molecule. Okay, so जब भी हमारा metabolism होता है, and when we get these protons, this increased protons will be generating a membrane potential called as proton motive force. Now, this proton motive force will enable the so-called opening and closing of this. You can see these two tubes here. One is the inlet and the other one is the outlet. Okay. So when this is opened up, then you have the movement of protons. This movement of protons now drives the rotation. How does it drive the rotation? Let's have a look at it. Yeah, as you can see here, this one is empty, which means that you have a binding space for a proton. So जो भी proton अंदर की तरफ आ रहा है, उसके लिए you have a place to bind that H. So when the H binds here, the conformation of backward protein changes, because of which it is going to leave one proton. Okay, so there is a reduction followed by a simultaneous oxidation happening in the entire complex. This makes the rotor to move. Again, we have a reduction. See, we have one proton entering from this and one proton taking an exit from here. So this way, the forward and the backward proteins always change their conformation, and that leads to pumping of hydrogen ions inside. So when this pumping happens, one thing you have to observe is that when this motor is rotating, you see the stock, which is made up of gamma, delta, and epsilon, also is like carrying forward the rotation force downward. Okay, so now this torque that is coming downward is sufficient enough for coupling. So what is, you can see here coupling. So one of the ADP molecules will be coming and will be sitting in the alpha beta complexes. Okay, and when rotation is happening for like 3 to 4 times, depending upon what you're looking, you have a phosphate molecule which will be binding to a diphosphate. So ये दोनों को binding के लिए बहुत energy चाहिए, and that energy is coming up through mechanical conversion. So what are we observing here? We are seeing that the chemical gradient is leading to generation of a mechanical force. Okay, so here your mechanical force is being carried downward, and this mechanical force is again useful for providing the energy for coupling of a phosphate to a diphosphate, leading in the generation of ATP. Okay, so यहाँ पे हो गया. Then look at the next one. So यहाँ पे भी already process हो रहा है. So this way, these rotations keeps going until the gradient becomes equal on both the sides. And this way we have generation of ATP molecules inside the mitochondria. Okay, and for this reason we are saying mitochondria to be like this तरफ जितना भी ATP आता है, they dominates all the so-called ATP generated different parts of the cell, and that's the reason why we are considering mitochondria as the powerhouse of the cell. Okay, so this is about your rotary motor that is available in your bacteria में भी एक छोटा सा motor है, mitochondria में this is what we have seen in mitochondria. The same sort of setup is available in the chloroplast also. The chloroplast has C elements being varied. C elements जो है वो अलग है chloroplast में, जिसकी वजह से वहाँ पे जो utilization होता है that will be lesser. Okay, in mitochondria probably they will be using 4 protons for every ATP production to happen, whereas in chloroplast they will be using only 3 protons. Okay, and in case of bacteria, the proton number vary. So that's about your so-called rotary motors. In case of eukaryotes, now let's look at the rotary motors in case of prokaryote. We are going to look at the complex of rotary motors that is available inside of so-called flagella, bacteria with flagellum. Okay, so flagellum को function क्या है? It takes parts in 2 different functions. One is locomotion and the other one is sensing. Okay, these proteins that are available here, they are also equally important for sensing. Okay, so locomotion के लिए this flagella will be completely dependent upon this lowermost segment of the membrane, which is composed of different rotors. Okay, and these different rotors are interconnected with each other by a central rod. The rod is in conjunction with these proteins called as flea proteins that are required for generating the power using the chemical gradient available here. Okay, they don't just generate the power for chemical gradient, rather they also determine the direction of so-called rotation. In rotations we have 2 rotations. एक तो clockwise rotation है और एक anti-clockwise rotation है. दोनों तरफ bacteria का flagella rotate कर सकता है. So when these rotations happen, automatically you see a whirling motion exhibited by this entire whip. Okay, so connecting the so-called whip and the basal body, you have a hook. The hook is also made up of the proteins, which are nothing but self-assembling complexes that are made up of a protein called as flagellin. Okay, so from here itself, the rod will be extending further into this. This will be getting connected to the flagellin, and the flagellin starts to do self-assembly with respect in conjunction with this hook, and this way you have an entire base being established. Okay, so what are the different components that usually found in bacterial flagellum? We see we have different membranes. Okay, so you have to remember the components. The first component will be a rod, because based on this rod only you are going to have different arrangements. Okay, so 4 rings available at the lowermost lipid part. The first one is the C ring, the second one is the MS ring, followed by we have a peptidoglycan ring, followed by we have a lipid ring, L ring. So 1, 2, 3, 4 rings on common. You see, you may have additional rings also. So these 4 rings, the first 2 rings will be having the entire machinery. Okay, so what are this entire machinery that I am talking about? One is flea proteins, which actually takes part in switching of the bacterial motion, and then we have more proteins, MOT proteins. These more proteins will be like moving up and down, and this generates the concentration gradient across this, and this gradient is utilized by the rod, central rod. And the central rod, as this is moving, the other rotors generate, and all of these rotors generate at once in the same direction. Okay, so these are basically like acting as shafts and gears, etc., etc. Okay, so this energy is generated in the upward fashion, which is which is again carried ahead to the hook. The hook also rotates in conjunction with the rings, and these rings will be deriving their power from two different ions. One will be sodium ion, and the other one will be potassium ions. Okay, so both of these ions will generate a electrochemical gradient, which usually will be approximately like around 150 millivolts. Okay, and this gradient is suitable enough for the bacteria to take part in swimming process. Okay, so there are, as I said earlier, there are two different types of proteins: mode A, mode B, flea. More proteins are required for motorized activity of or the so-called generation of the rotational forces for all of these rotors, and flea proteins are required for the direction. Whether this has to take part in a clockwise direction or an anti-clockwise direction, that will be set by the flea proteins. Okay, so if you're going to get the rotational force that comes, the rotational speed that comes out, the bacterial flagellum is going to rotate at a speed of 1000 hertz when it is going in a clockwise fashion. मतलब ये जब रोटेशन क्लॉकवाइज फैशन में होता है तब रोटेशन का स्पीड कितना है 1000 hertz है. And this is the reason why you tend to see the bacteria moving at a great speed, like it moves at approximately like 50 times or even higher than its body length. बैक्टीरिया का जितना भी बॉडी लेंथ है 1 second में it is going to cover 50 times or even higher than that of its own body length. Okay, and that's how these microscopic creatures tends to move faster compared to the higher creatures like us with respect to the body lengths. Okay, so let me just demonstrate a small video so that you can understand it better.
Excuse me, sir.
Yes, tell me.
So my internet connectivity was not stable, so I couldn't able to mark my attendance. Can you mark my attendance now?
Yeah, I'll mark it. I'll mark it. Okay, don't worry.
All 834.
Okay. So as you can see here, this is how your rotors are being arranged. This is your so-called hook region. This entire complex is called as basal body. So you can see these so-called functioning of these is completely dependent upon the so-called membrane gradients that are available here. So this is the proton gradient that we were talking about. So same oxidation and reduction reaction was happening. And if your advertisement, give me a second. So look at the way the flagella is actually moving. Look at the way the flagella is moving. Now this is a clockwise direction. So this kind of a direction is exhibited when the bacteria has to swim. मतलब एक जगह से दूसरी जगह तक जब जाना चाहते हैं bacteria, at that point of time, the direction of rotation is clockwise. Simply remember that. And when does the bacteria exhibit an anti-clockwise direction then? When a bacteria is settling down at a particular place and when it is sensing danger and when it is sensing food. At that point of time, the rotation is going to be in a counterclockwise direction. Okay, counterclockwise direction, majorly the bacteria uses it for so-called sensing of nutrients. Okay, and sensing of dangers as well. So there are two different directions. This direction the bacteria will be employing when it is swimming. So when the bacteria is going in a clockwise direction, the power generation is going to be a bit higher. Power consumption is also going to be higher because in swimming pattern, the so-called the filament is going to rotate at the speed of 1000 hertz, whereas in counterclockwise direction is going to be a lesser. Okay, so they again have different gradients. So people expect that when it is swimming in a clockwise fashion, it's going to utilize both hydrogen ions and sodium ions, whereas when it is going to swim in counterclockwise direction, a little bit lesser amount of sodium ions are being utilized, and that's the reason why the rotation speed is going to be lesser. Okay, so you see this is a rotation. Okay, this is a rotation. and the same rotational force is been going ahead across the entire so-called flagellum. Okay. This way, the flagellar motors do take part in the working of the flagellum. Now, coming to the other part, the so-called linear motors. So, if you're going to look at the linear motors, oh yeah, this we have missed. Sorry. Okay, so coming to the types of bacteria based on the availability of the flagellum, we have 6 different types of bacteria. Number one is atrachus, which means there is no flagella available on this bacteria. They don't have any moving structure at all. Okay, so these kind of bacteria are atrachus, which means no flagella on the body of the bacteria. Then monotrichous, which means there is just one bacteria which is available at one end on the bacteria. That's called as monotrichous. Then lophotrichous, which means you have different flagella and many numbers of flagella are just available at one pole of the cell. So that's called as lophotrichous. Then amphitrichous, which means you have only two flagella, but two flagella are available at two different junctions. Okay, that's called amphitrichous. Then peritrichous, which means that the entire body of the bacteria is occupied by flagella. There are many flagella, all of these flagella are spread over the entire body. Then amphileucotrichous, means on the two different regions you have many flagella available. So this is amphi. Amphi matlab you can see this is also amphi, this is also amphi. यहाँ पे सिर्फ एक एक साइड में एक यहाँ पे tuft है. You have many flagella available as tuft. Okay, so mono, this is also, both of these tends to be like single pole, but here you have only one, here you have many. Okay, so this is mono, this is lopho. This is amphi, this is amphilopho. Okay, and then this is peri. So trichosity usually indicates about the availability of flagellum. So trichous is the term that we are using for availability of flagellum. So remember that these are the different types of flagella that can be seen in the nature. Okay, now coming to the linear motors. So what does this linear motors do? Linear motors takes part in the so-called activity where the energy consumption happens. Okay, यहाँ पे हम क्या देख रहे थे? यहाँ पे हमारा energy generation हम खुद से I mean this is, they are doing the process on their own. Okay, one way they are either giving you an output in the form of ATP or they are giving you an output in the form of a movement of certain so-called structural component. Okay, so in both the cases they are dependent upon the voltages, but here in case of linear motors there is nothing to do with the membrane voltage. Okay, then what are we dependent upon? We are dependent upon the ATP that is generated because of those molecular motors. Rotary motors के वजह से जितना भी ATP generate हुआ है या बाकी reactors के वजह से जितना भी ATP generate हुआ है वो ATP का इस्तेमाल करते हुए they exhibit mechanical function. Okay, so we are going to study 3 different types of motors. One will be kinesin, other will be myosin, and the other one will be dyneins. So we have 3 different molecular motors that we are going to study. All of them have a common structural function, components. As you can see here, they have a motor domain which is indicated in slight yellowish color, followed by you have a central stalk. The stalk can be flexible, and this is usually made up of polypeptides, different types of amino acids. Okay, and in some cases you may have light domains which is required for binding to the cargo or to the so-called other elements, and so this binding happens with different proteins, and in some cases you may not have so-called light chains. So this way a typical linear motor will be having motor domain, light chain, central stalk. Okay, now let's start with the so-called cytoskeleton. So cytoskeleton is very crucial, very important for these motors to work. ये motors काम करने के लिए या motor domain काम करने के लिए आपको cytoskeleton की बहुत जरूरी है. So on cytoskeleton we have 2 different things. One are microtubules and the other one are microfilaments. So microfilaments are required for one category of proteins called as myosins, which you find in muscle. And these 2 are dependent upon the microtubule portion. So myosin is made up of a protein called lactin and so So microfilaments are made up of a protein called as actin, and microtubule are made up of a protein called as tubulin. Okay, so actin and tubulin tends to interact with the protein's motor domains, and here the interaction is going to lead to so-called breakdown of ATP molecules, and the energy that is released out is being utilized for the mechanical activity. Okay, so we begin with the myosin linear motors. So what does this myosin linear motor do? So in your muscles, if you're going to look at the cytoskeletal arrangement inside a muscular cell, the so-called microfilaments are arranged in superficial layers, different superficial layers. Okay, and these different superficial layers will be taking part in overlapped sliding activity. Okay, matlab, just like the way this boat is rowing across this so-called river, water body. Okay, your myosin will be enabling the movement. मतलब यहाँ पे हम बोट को क्या कर रहे हैं? बोट को हम आगे की तरफ बढ़ा रहे हैं. Let's say this is your myosin, and let's say this is your so-called actin microtubules. So when these two comes in interaction, and when the energy is being so-called utilized, this will be making the so-called microfilament to slide. Okay, and that sliding will bring out a change in the cytoskeletal composition. मतलब composition मतलब आपका जो cytoskeletal volume है वो constrict हो जाएगा, कम हो जाएगा, जिसके वजह से क्या होता है? That will be leading to shrinkage of the entire muscle, and we call it as contraction. Okay, so contraction is a process which is dependent upon the binding of your myosin to the actin. Okay, actin is available on the microfilament. So when the myosin heads binds onto the actin, you have a breakdown of energy from ATP to ADP. So when you get this conversion happening, then that leads to sliding of your so-called actin filaments. So this is called as a power stroke. Let me just demonstrate it through a small animation. Again, let's get back to YouTube. Yeah, this is visible. Okay. So as you can see here, yeah. See, whenever the energy is being moving outward, you can see the actin filaments are actually, this is your actin filament. Okay, ये आपका actin filament जो भी है, that actin filament is being dragged upward. Okay. Now that was a forward stroke, and now this is a recovery stroke. So recovery stroke में क्या हो रहा है? ATP, दूसरा ATP आके, it's going to bind to your myosin. So now this myosin is moving in such a way that the mechanical force will be splitting up your so-called triphosphate to diphosphate and monophosphate, and that takes part in recovery. Okay, so here you can look at the overall muscular components. See, ATP breakdown होने के वजह से क्या हुआ है? The binding of this so-called myosin with the actin has happened, and that's actually pulling your cytoskeletal elements. So now these microtubules, you see, they are being dragged up, and that leads to contraction. So that is called as power stroke. Okay, you can have a look at it once again. ATP hydrolysis हो गया है, hydrolysis होने के बाद phosphate will be leaving, and the binding of this myosin pockets into this is required. So once this is gone, the so-called cytoskeletal elements are dragged. Okay, and so this way you have a contraction and a relaxation. मतलब ये आगे की तरफ बढ़ेगा और फिर पीछे जाएंगे. So this आगे पीछे मतलब the forward and the backward motion is completely dependent upon the breakdown of ATP, and that is who's doing that? That is being done by your so-called myosin heads. Okay, so the myosin will be trying to fit into the actin filaments, and it will be able to drag it. Okay, so that forward and motion leads to contraction and relaxation. So that's one thing. Now coming to the second kind of motion that we actually come across. Let me just share. मतलब साथ ही साथ कर लेते हैं. Yeah. So this is the second kind of motion that you actually find. So these are the microtubules now. ये stout है, काफी stout है. Microfilaments thin है, made up of actin. This is made up of tubulin. So you can see there are a lot of microtubules available inside your cell. Okay, so this is your cytoplasm. This is just an animation. So in your cytoplasm, on different microtubules, you can see there are different kinds of trafficking being done. So transportation being done. So how does the microtubule help in transportation? And what are the proteins that are involved here? We call them as kinesins. Okay, so you can see the general structure. This is your motor domain, this is the stalk, and this is your light chain. So the light chains are bound to the cargo through strongest interactions. So the cargo is actually coming out from your Golgi. Golgi apparatus का काम क्या होता है? Sorting and so-called trafficking, packing, sorting, and trafficking. Okay, so trafficking, it will be deciding where and how it has to get trafficked. Matlab, the proteins which can actually do the transportation, they can do the job in cytoplasm, in the cytoplasmic medium, or there are these combination of motors that actually can do this transportation. So this is going to act as a pulley, just like the way we have pulleys or the porters available in railway stations or bus stands. Okay, you see the same sort of a labor also available in your cell, and this is your labor. So we call this motor as kinesin. So what does this do? That will be linking onto your cargo through these light chains, and your so-called motor domain is flexible. Okay, so let's look at the working activity. You see, they are actually working on the microtubule. So how do they do that? For every step that they are actually bringing out, see, these are the two legs. So let's say these are the two legs. So this is one, this is a motor domain, and this is also a motor domain. So each motor domain will be breaking down ATP. Again, just like the way वहाँ पे आपका माइसिन में जैसे ATP break हुआ था, वैसे यहाँ पे भी ATP break होता है, जिसकी वजह से this conformational, the stock will be undergoing a conformational change that makes it move its limb forward by 80 angstroms. Okay. So we have an 80 angstrom motion coming this way and this way. Okay. So carrying the load, the kinesin motors will be moving across the so-called microtubule. You can see the one guy is moving here, and you can see other guy is moving here. And this way, in the entire cell, you can see there are a lot of trafficking happening. You see, these are all the vesicles that are being trafficked. Okay. And that's how the so-called kinesin motors are going to work. Now, coming to the third category of motors called as dyneins. So dyneins, what do? They don't, unlike myosin or unlike kinesin, dyneins can't function independently. They have to function in the group. मतलब ये छोटा सा linear motors है जो इकट्ठा करके they are going to bring out a big change. Okay, so they themselves will not be doing a job on their own. Rather, they are interconnected to several other structural proteins. And these all structural proteins are again in conjunction with your so-called cytoskeletal elements. And through the cytoskeletal elements, you are actually going to generate a mechanical activity. So how do we do that? Let's have a look at it. So now this is a eukaryotic cell. So this is a sperm actually swimming. Okay. So when a sperm is swimming, you can see here also it is utilizing a flagella. Okay. So in eukaryotes also the locomotion can happen in single cellular cells. The locomotion can happen using cilia or flagellum. Okay. But how is this flagellum different than a bacterial flagellum? The bacterial flagellum is exhibiting a rotary motion. Okay, the forces there are rotational forces, whereas here the forces are not rotational. Okay, so what kind of force is this? What kind of a mechanism is it? It's kind of a wave mechanism. The wave is being generated, and if it is cilia, you tend to see that as a vibrational motion, and if it's a flagellum, you tend to see it as a wave motion. Okay, which means that you have a forward and a backward stroke being generated because of activity of certain linear motors. So how does this happen? Just have a look at it. So this has a crest and a trough, right? Which means that you have two different components acting on the opposite ends. Okay. So let's look at the cross section of this flagellum. So you're going to have all of these structural proteins that takes part in establishment of this flagellum. Center में आपका rod है, and all of these are interconnected with your cytoskeletal elements. So in between, you can see these white structures. These structures are your small linear proteins, which are showing a synchronized activity. Okay, so this is the cross section of your flagella in a eukaryote. Okay, so you can see these dyneins basically exhibit a mechanical activity on hydrolysis of ATP. Okay, so let's look at the animation. Yeah, they are binding to the structural proteins and they are pulling them. This is called as the power stroke. They are releasing back. Recovery stroke. So they get locked up, they pull up, they unlock. Okay. So this entire microtubule track that is available in association with them will be exhibiting a wave motion. Okay, and after certain motion, they tend to get inhibitory signals because of which they don't act in a synchrony. And when they don't act in a synchrony, they don't exhibit any power motion. मतलब ये जो cytoskeletal elements से जो binding है, यहाँ पे देखिए cytoskeletal elements से binding हो रहा है. So this is called as inhibitory phase or recovery phase. So there is a power stroke where you tend to see the binding of both of these so-called dynein fragments to different microtubules. Okay, and that takes part in movement of microtubule entire body. And there is a recovery stroke or a relaxed stroke where you tend to see an inhibitory signal that doesn't make these pockets to interact with the microtubule. So इस तरह अगर आप पूरा microtubular arrangement को देखेंगे, if you are going to look at the entire cross section, see you are going to get a activity generated at one side and the other side this is free. Now here activity हो रहा है, यहाँ पे free है. Again just have a look at it. When they are moving, this is free. So when they are moving, you see a wave generation happening in this side. And when they are moving, these are in inhibitory state. So you don't see any wave generation happening there, and you see a wave generation coming from this side. So that's how this entire body is exhibiting a crest and a trough. Okay.
Yes.
Now these are so when these are functioning, they will be ideal. When these are functioning, these will be ideal. Okay. So they exhibit both power stroke and recovery stroke. So when this is under power, when this is under recovery, when this is under recovery, that is under power. Okay. So that way the flagella will be exhibiting a wave motion, and this is how the mechanical activity is attained, and this is how the so-called the locomotory activity, whatever has to happen with respect to cilia or the flagella, will be exhibited. Okay. So that's about your molecular motors and machines. So the same has been given on your presentation also, but it is better to demonstrate it through video. So I have to utilize some media component. I'll be sharing the links with you all. At the same time, I'll be sharing the PPT with you all. So you can go through them, okay, and prepare accordingly. And I'll be sharing a document with you all where you have to start writing the so-called answers to that. Okay, I'll be giving you a deadline. So within the deadline, try to submit it. Okay, that's your assignment. It's a written assignment. So first we'll do the written assignment, and next week, probably at the end, we'll be having a test also. So prepare well. Okay, the test will be of chapter number 3 and chapter number 4. Shubham, I have marked you. Don't worry. Okay. So thank you all. Thank you all. That's all for the today's class.
Excuse me, sir.
Yes, tell me.
Sir, tomorrow we have the class.
No, tomorrow I'm not sure. We may not have. Tomorrow 8 o'clock है क्या? Kal 8 o'clock. No, tomorrow we don't have a class.
Okay, sir. Thank you, sir.
Yeah, tomorrow you don't have to come.
सर वो टेस्ट मतलब क्विज होगा ना?
बिजी होगा. अभी पहले मैं असाइनमेंट पोस्ट करूंगा, उसके बाद क्विज के बारे में सोचेंगे. असाइनमेंट आप लिख लीजिए. ओके.
ओके सर.
So Shubham 834, I have marked you already.
Sir, 784 present.
784.
Yes, sir.
Yeah, it is marked.
Sir, 818 also.
Done.
Thank you, sir.
818 done. Okay, thank you all.
Thank you, sir.
Thank you, sir.